It's a great pleasure to be here to discuss the background to cloning. Why did people first start thinking of nuclear transfer? How did the technologies develop, and how did we ultimately use that technique to produce Dolly? Before I move on to briefly discuss some of the implications and limitations of the cloning procedure. The first point that it's essential to make is that this sort of project is a very large project which depends on a considerable group of people with different expertise. I've chosen to pick out this small group to start with because these are the people who are most closely concerned with the nuclear transfer technique. Mention's been made already of Keith Campbell, who put the biology of the cell cycle into the project, which was, as I'm going to demonstrate, was really essential for our success. I'd also pick out Lorraine Young, who led the work on imprinting genes, which I'm going to refer to. It was a major part of the research. But as I emphasized initially, this is research which requires a considerable variety of different expertise and therefore a considerable number of people. And these, I hope, are all of the people who were involved throughout the project. Top left, the group concerned with nuclear transfer. Bottom left, colleagues who were involved at different points in the project, sometimes growing cells, helping with the surgery, a variety of things. The middle group, Tim King and Susan Ryan are both veterinary surgeons. Tim had care of the animals at some points in the project. Susan carried out the detailed histopathology on Dolly after she'd been euthanized. The other people in that group there were concerned with looking after the animals, particularly during their surgery. Many of you will recognize John Clark and his colleagues who were interested in having the opportunity to make genetic change in the animals. They were involved in the prior project to add genes into sheep with the objective of getting them to produce in their milk proteins that might be used to treat human disease. The original objective of the project was to establish the opportunity to make precise genetic change by having cells in which we could make the change before we did nuclear transfer. We didn't originally set out to clone adults. We set out with that different biotechnology objective. I've mentioned Lorraine Young and her work on imprinting genes already. Below that group, the bottom group, bottom right-hand side, we used a model system to study what became known as the large offspring problem, working with a group in Aberdeen who were led by John Robinson, and those are the rest of the people who were closely involved in that project. So as I said, a great variety of expertise, a large number of people without whom this project couldn't have been completed. On a number of slides, you'll see that there's a date in the top left-hand corner, which is roughly when an event took place or the environment that I'm referring to was predominant. So it's as far back as the middle of the 19th century when people were thinking about the basic question as how does the single cell of an embryo give rise to all of the different tissues of an adult? A question to which we still don't have all of the detailed answers. But as you'll appreciate, at that time when they had much lesser understanding of the basic biology, of molecular biology and so on, they had even less research to work on. It was really largely a thought experiment. And the second one related to it, is it possible to change cells from one tissue type to another? And Weissman was a person who had a great influence on this sort of thinking and analysis. And I've chosen one particular quote because it underpins the original approach to nuclear transfer. He argued that it must follow that during segmentation and subsequent cell division, the nucleoplasm will enter upon definite and varied changes which must cause the differences in the cells that are produced. That seems perfectly reasonable. The simplest hypothesis would be to suppose that at each division of the nucleus, its specific substance divides into two halves of unequal quality, so that the cell bodies would also be transformed. Now this phrase, unequal quality, is rather sort of vague and general. What I think it was often taken to mean was that there was loss of some of the material in the nucleus, loss of some of the genes. So that, for example, if a cell was going to produce muscle, it would retain those instructions to produce muscle, but not those for the production, for example, of nerves. There's no indication as to how this could possibly take place, but that I think is the way that this was understood at the time. And you can easily see how that leads to experiments related to nuclear transfer, because if you took a cell from an adult, a differentiated cell from an adult, it would have lost the mechanisms to control development of lots of different tissues, and so wouldn't be able to support development to term. And that's, I think, the original idea why people thought about nuclear transfer. Of course, the current technologies just didn't exist, and they used ingenuity to ask at simple early forms of this experiment. Shown here, one very early one with sea urchin embryos, which were split. And you can see from the diagrammatic representation that it seems that at the 4-cell stage, each cell retained the ability to form an entire offspring. So there'd been no loss of material at that point. A very famous experiment by Hans Spemann, this is a photograph at the right there, excuse me, of his lab book. What he did was to take the eggs, this time of salamander, and tie a ligature around the cytoplasm in order to restrict the nucleus to one part and have a different part, which was anucleate. And he allowed the cell with the nucleus, the fragment with the nucleus, to divide. You'll see that the other half didn't fragment, so that division was dependent upon the presence of a nucleus. And it divided to the 16-cell stage when the ligature was released and a nucleus was allowed to migrate across into the anucleate fragment. He then tightened the ligature again and allowed development to continue. And I hope you can see in the diagram on the bottom right, excuse me, that there are 2 salamanders there. So the answer was that no material had been lost by that 16-cell stage. Perhaps as importantly, that experiment is published in 1928. Perhaps as important as that, in 1938, he described what we would now recognize as a nuclear transfer experiment, but the techniques, the equipment were just simply not available. The first people to actually attempt this and succeed were Briggs and King, working in the United States with one species of frog. But a few years later, it was John Gurdon, working in this country, who really took the technology further forward with a different species. A very famous photograph there where you can see all of the offspring which were produced by nuclear transfer. And you'll see that he used a color mark to distinguish the offspring produced by nuclear transfer with a nucleus from a lighter colored animal from the animal which had provided the eggs. These could only have been formed by nuclear transfer. By this time, it took a while to develop the technologies. Obviously, adults had been produced by nuclear transfer from tadpoles, and tadpoles had been produced by nuclear transfer from adults. And I think to most people that would be convincing evidence that development did not depend on loss of DNA, loss of genes. But it was what I'm sure must have been a very frustrating result for him, that despite this and the importance of his conclusion, no adults have ever been obtained by nuclear transfer from adults. And I think that's still the case. And I'm sure he's tried very hard. And at this time, because of the failures, I think people did not expect to be able to reprogram adult cells, either in the sense of nuclear transfer or in our modern jargon use of the word, to change the phenotype of the cell from one type to another, because they regarded them as being rigidly fixed. You can see in the graph there on the right-hand side two lines. The lower one is the American work with Rana pipiens. The right-hand one is the work with Xenopus that was done by John Gurdon. And you can see that he was able to get the cloned animals to develop further. There had been changes in technique as well as use of different species, and it's simply not clear which of these factors was important in providing the greater success. There were a number of technical problems which had to be overcome before similar experiments could be done in mammals. And Steen Willardson spent some time, first of all, splitting embryos and developing the enabling technologies to encase the manipulated embryos so that they could be put back into foster mothers and still have a chance of developing. Recovering embryos is, of course, necessary expertise if you're going to do this sort of research. And he was the first person to produce reproducible nuclear transfer with ruminants. You can see here just a representative photograph from his Nature paper of cloned embryos, and then bottom left, the cloned lamb. There was a different type of experimentation which also gave underpinning information to this question, and that was basic studies of the mechanisms that regulate differentiation. And it's a shock to discover that this was only 30 years ago. The conclusion has been drawn that all cells have the same DNA, essentially, and that formation of different cell types must involve changes in expression of genes. So a number of people were looking for the factors which regulated these changes. And one of the ways of approaching this was to have fragments of cDNA from appropriate tissues which were injected into cells to try to locate regulatory factors. And differentiation to muscle fiber was a system that was studied in this way. So there was a critical paper published in 1987. And it was, when I first thought about this, it was a real shock to realize that it was only then the concept of transcription factors and the identity of transcription factors was only then beginning to become clear. We're so familiar with their use now that that was a surprise. So what this shows is the effect of introducing one of these candidates, MyoD, into 3 different cell types, some mesoderm, from which muscle normally develops, skin, and liver cells. And you can see from the representation of the tick or the cross that this single factor was capable of promoting the differentiation of the cell along its normal lineage from mesoderm through to muscle fiber. On the other hand, if the same protein was established in skin cells or liver cells, that effect could not be achieved. And so it perhaps gave a rather negative view of what could be achieved with a single transcription factor at least. So I've just, in this slide, just summarized the same points that I think some of which we would still hold, that mechanisms that control development are complex and rigidly fixed, and it is difficult to change them, but we are now learning how to do that. So the question then, why was it that we were successful in nuclear transfer? And the bulk of the work was carried out between 1993 and 96. A variety of experiments had noticed effects of the cell cycle stage of donor cells and the recipient cells on subsequent development. But there had been no really systematic analysis of these relationships. Keith Campbell was able to do this particularly well because he understood the regulatory mechanisms in cell cycle and was familiar with the research techniques. He earned his PhD from work done at Sussex, trying to identify key proteins from the cell cycle. And on the basis of his expertise and knowledge, we carried out the first systematic analysis. So just to remind those of you who are not familiar with this subject of the nature of the cell cycle, in this diagram, you can see that it's going round clockwise as we look at it. And there are a number of key stages. M phase, metaphase, where the chromosomes are aligned and then division can take place. The G refers to gap, so that there's a first gap when the cell cycle continues or it's initiated, before you get into S phase, where DNA and lots of other proteins are synthesized, and you get to the second gap when the cell begins to organize itself ready for mitosis. And a key phase is M phase, whether it's in a mitosis in meiosis or in a somatic cell. it's easy to imagine, even if you don't understand the detailed mechanisms, that if you have an oocyte which is at metaphase 2, and you have a donor cell which is in S phase, for example, and you mix the two, that you may well get an unfortunate outcome from the point of view of nuclear development. In most of the nuclear transfer, but not all, nuclei are transferred into an oocyte as it is recovered soon after ovulation, when it's at the metaphase 2 of meiosis. And it has acted upon the chromosomes to condense them on a spindle prior to ovulation. And so it's not a surprise that if you put a nucleus in at this stage, the same thing happens to those chromosomes, that nuclear membrane breaks down and the chromosomes are condensed. And that's perceived as offering an advantage because it's exposing the chromosomes to the other factors which are present in the cytoplasm. But potentially a disadvantage in that among those factors are those which promote DNA replication. And as I'll show you in a moment, it proves to be absolutely critical to transfer in a nucleus which is awaiting DNA replication. I mentioned this experiment last night in the public lecture and discovered subsequently that the then Spanish student who did this work was actually here today. So it's a great pleasure to see you here, Pedro. And you may be slightly surprised to still see your data being published. So this is an experiment which shows the effect on development to blastocyst. It's a rather limited measure of development of embryos produced by putting nuclei, the 4-cell stage, into oocytes and culturing them. This is showing percentage just for the ease of reading this and so on. The experiment was replicated 3 or 4 times. The 43% and the 60% are not significantly different. So it seems that the G1 stage, the first gap, is particularly appropriate from the point of view of the donor cell. There's clearly a profound influence of the metaphase 2 oocyte in that it is not suitable for cells which have reached the S phase. One probable reason is that they've already begun to initiate DNA replication and are triggered to reinitiate replication, leading to a situation of aneuploidy and subsequent failure. It's only in this situation where there is an appropriate match between the oocyte which will promote nuclear membrane breakdown and a nucleus which is awaiting DNA replication. It was a bit of a surprise to get the same success with the early S. It may be that the time that was chosen was relatively early on and could be tolerated. So just to remind you then, what we have shown in that experiment is if you put nuclei in here, at the stage when they're awaiting DNA replication, it can be very successful. But if you put in nuclei at a different stage of the cell cycle, it will lead to abnormal development. The way in which we overcame this was by recommending the use of and selecting the use of oocytes which had advanced into S phase. Because they'd lost the ability to stimulate DNA replication, we christened these universal recipient, because that environment was suitable for a nucleus at any stage of the cell cycle. We were able to look back with a greater understanding of the cell biology and to suggest that the reason why Willardson was successful in his work was that he was using oocytes which were relatively aged. They were 40 hours after gonadotrophin injection when they would be expected to ovulate after 24 hours. And at this stage, they are known to be readily activated. So it may be that he was using oocytes which were in S phase without really being aware of it. He was using what we'd come to christen universal recipients. So on the basis of this understanding, we were able to transfer nuclei from differentiated cells. First of all, embryonic stem cells, well, sorry, not embryonic stem cells, embryo cells which had been cultured with the hope of obtaining embryonic stem cells. a very early differentiation, subsequently from fetal tissue, day 35 of pregnancy. Clearly the effects of cell cycle stage in both egg and the donor cell, with the optimum combination being to have an oocyte at metaphase 2 and the donor cell awaiting DNA replication, and the alternative use of a universal recipient. There was one other factor that we introduced, again Keith introduced. You'll notice up here a phase which has not been mentioned, G0 or quiescence. It was very difficult to work with the blastomeres of the early embryo because they do not respond to the normal checkpoints. The way that Pedro did the experiment we've just looked at was by observing them as they passed through mitosis and then waiting a set number of hours until they were presumed to be in a particular cell cycle stage. Once they get past a certain stage, cells will respond to serum starvation by exiting the cell cycle and becoming quiescent. A huge improvement of convenience, if you think about it. You can starve them for 3-4 days, they all enter quiescence, and you can leave the dish on the bench and take the nuclei, the cells out as you want them. None of this watching to see when they divide. So there's no question it was a great convenience to us when we began to use this with the fetal tissue and with the mammary tissue from which we obtained Dolly. And so putting all of those things together, this was the basic mechanism by which we were able to obtain the first adult clone. You'll recognize it, of course. Like Gurdon in the experiment that I showed you with the frogs, we also had color markers because, as you know, Dolly is from a cross which has a white face. All of the other sheep involved were black-faced, and that name is meaningful. If there'd been any black-faced genes in Dolly, she would have had a colored face. There's been quite a lot of discussion and debate as to whether quiescence is beneficial. Historically, there's no doubt in my mind that it was important for us, but that's a different question. And one of the groups, the Ruakura group in New Zealand, for example, did quite a substantial trial comparing serum-starved G0 with cells which were picked up soon after they divided as a way of getting G1. And there wasn't a consistent result either way. They seemed to me, looking at the data, to be very, very similar indeed. The paper was written with a certain degree of skepticism as to whether G0 was useful or not. Under those circumstances, I was particularly interested to see this paper which came out, I think it was in June this year, where that group had looked at the chromatin in the donor cells and in developing embryos. And we don't have time to look at all of their data, but I recommend the paper to you. The full reference is at the top. Summarizing it, they showed reduced levels of particular polychrome proteins in the cells that were quiescent, and hypomethylation of both DNA and some histones. After nuclear transfer, one of the histones still remained hypomethylated in both the inner cell mass and the trophectoderm, in other words, the whole of the blastocyst. And they associate these differences with relaxed chromatin. And so they suggest that it's because of this that it's easier for the factors which are going to bring about the reprogramming, it's easier for them to have access to the DNA. So it would seem as if they're advocating this as a mechanism by which G0 confers a benefit. But what about the details? What about the efficiency of this procedure? This is French data which I've replotted, comparing the survival of embryos produced by IVF, embryo transfer, nuclear transfer from fetal tissue, and nuclear transfer from adults. And you can see, much as in the original frog data, as you take nuclei from progressively later stages of development, the success rate drops. You'll also notice that this is not to scale, of course. Carving is after 280 days, I think. Is that right? There'll be somebody in the audience who will tell me if I'm wrong. How long's pregnancy in cattle? Yeah, right. So I was right. You'll see that there's continued loss of fetuses throughout pregnancy, but notice that break in the scale, and a greater loss the later the stage of development from which you took the nucleus. This is 10 years old, and I'm not sure whether any other species or crosses have actually been cloned, but you can see a great variety. The outstanding exceptions are the primates, for which we only just may be getting now a procedure which is reproducible and successful. Early results showed that if you applied the Roslin procedure to human or non-human primate oocytes, that you got minimal development. And relatively recent investigations have shown that they are activated prematurely. If you handle the eggs in the sort of way we handle ruminant eggs, they are partially activated, which is not good. But then when you transfer in the nucleus and try to completely activate, you get an inadequate response because your response has already partially taken place. Since then, the procedures have been modified to overcome that disadvantage, and they are now producing blastocysts from which they can derive embryo stem cells, very high success rates. There's one paper which is really not very well known. It deserves to be much better known than it is. And you'll see the name of the lead author at the bottom there. It's data which was derived from work done by the Granada company. When I first visited them, I was told that they were trying to prepare 1,000 clones, as part of a trial. And knowing how difficult it is, I had to be picked up off the floor. I mean, I was astonished. Why would you do this? But it was a very sophisticated design. They inseminated large numbers of heifers with semen from a small number of bulls for one group. So those are all full sibs. They took embryos from donors which had been superovulated and inseminated by some of the same bulls, where you have, again, full sibs, but also half sibs to the top group. And then they did nuclear transfer, from some of the embryos. So you have then a clone plus full sibs and half sibs. So you'd expect that the clones would be the most uniform and that there wouldn't be a significant difference in weight between these different groups. Again, I'm not making time to show you the data, I'm just showing you the conclusion. The striking thing, and it was statistically very significant, was that weight was the most variable in clones at birth. There was a small effect of the embryo transfer. I don't think it was statistically significant. It was certainly much smaller. But even that had disturbed the development of the offspring. By 1 year of age, the results were different in that they were then, as you would have predicted or as you would have hoped, that the clones were the most consistent. So this is an effect before birth. So this makes it important to think, if you're using clones, that clones may be more variable than expected, than offspring from natural mating. And I'm glad I put in the caveat at the bottom, with the procedures which were in use at the time, which were essentially Steen Willidson's procedures, because he set up the work that's done there. So if failures reflect epigenetic effects, what are the mechanisms? When do they arise? What do we need to think of from the point of view of nuclear transfer and embryo culture? And Lorraine and her colleagues looked at imprint, taking advantage of knowledge gained by the group we were collaborating with in Aberdeen. And they had lambs that had been cloned in several projects. They took lung tissue soon after death, and the imprint was compared with that in parthenotes and controls. And they examined methylation of one regulatory element of IGF-2R. The work from the cultured embryos had shown that this was a gene which was changed substantially in animals that were unusually large, in fetus that were unusually large. The suggestion would be that if you have less receptor, which is involved in the removal of IGF-2 from the circulation, then you get a high level of IGF-2 and the stimulation of growth. And in a particular intronic regulatory region, there had been an almost complete loss of methylation in 9 of 12 cases. Since it was only 9 of 12 and all of them were clones, Lorraine suggested that there may well be other genes involved. So then, in making just one point regarding the use of the technology, we now know as a result of these experiments that cell function can be modified, but only inefficiently. Cloning has been used to make genetic changes in livestock, primarily to study inherited human diseases. The larger animals with perhaps physiology more closely resembling that of humans, being better disease models than mice. But there's no doubt that the greatest inheritance of the Dolly experiments was that biologists were made to think differently, and in one particular way. That experiment had shown that an egg can change cell type, and lots of people asked, are there other ways in which you can change cell type? And this is where we go back to the significance of transcription factors. Can we use transcription factors to bring about a desired change? And I'm not going to show you data, I'm just using a schematic just to show you that that is exactly what Shinya did in his experiments, which he'll talk about later on. And ever since I heard about that work, it seemed to me that those experiments are among the most important so far this century, and I think they're very likely to hold that status still at the end of this century. He will go on, I'm sure, to describe in more detail how pluripotent cells produced in this way can be used to study diseases like motor neuron disease. The particular point I wanted to make, because the idea was developed most strongly here in Edinburgh, is that it would be very useful to produce a library of selected cells ready for use in cell therapy. Craig Taylor, an immunologist at Addenbrooke's in Cambridge, has done calculations which suggest that if you do that, a library of 150 lines would provide a useful partial match for more than 90% of the UK population. Now, when iPS cells first appeared in the popular press, it was suggested that you could have personalized medicine. I just don't think we can do that. I have no idea how many new diabetic patients there are every year, but it would run into six figures, I'm sure, just in this country alone, and that's just one disease. We cannot afford the resources to produce patient-specific cells. Maybe Shinya will disagree, I don't know. But it seemed to some of us that a suitable library would be a much more appropriate way of addressing this. And in particular, we advocated that it should be viewed as a global task. You can sometimes have situations where, if you're looking for a cell, you can identify a person in a different country with the appropriate genotype, but not be able to get the cells because they wouldn't be available under appropriate, what would be deemed by us to be appropriate regulatory mechanisms. An intensely frustrating situation. So what we set out to argue was that a global scheme should be put in motion to produce a library within a variety of different countries, and it is now launched, by the way, with the aim in the end, probably 10 years or more into the future, of having a cell line which would offer a partial match to everybody in the world. And that does seem to me to be an objective which is well worth striving for. These cells could be used not only for cell therapy, but also for gene therapy, as it says at the bottom. I think I'm getting the victory sign. As it happens, just come to the end. I hope that I've given you the history of how we produce Dolly, and also taken the opportunity to explain why she's viewed by biologists as being the result of an extremely important experiment. Thank you very much.
So Ian told me before the talk that he's the proud father of 3 children, 5 grandchildren. I think one can also say that he's the father of an area of science, and we've had an inspiring insight into how he came down that road. So are there any questions? Anybody on the Twitter-sphere?
I want to ask you what you can tell us from reading the literature about the mindset that was prevalent around the time of the Briggs experiment. So you said that many people thought that muscle-specific genes would remain in a muscle cell and everything else would be thrown in the bin. So it seems therefore quite a brave thing to take a nucleus from a fully differentiated cell and put it into an egg. So did people think that they would just splurge and not get anything useful out? And how would that be viewed? How would early failures be viewed in that kind of context?
So whose experiments are you thinking of?
Gurdon and the people that went, the Americans.
Yeah, yeah. I think particularly the American group who really were applying a new furrow. It was very much an empirical, let's see what happens when we do this, which doesn't really help you very much. I don't think I know the answer to the question. Sorry, Ian.
More questions, I think. Sure. Sorry, down the front here.
It was such a nice talk. I just want to highlight or ask about your opinion regarding using transcription factors to reprogram cells. We don't much pay attention to the cell cycle when we introduce the factors. But as you mentioned clearly, a nuclear transfer experiment that plays a major role. Do you think that would also be important for the transcription factor mediated reprogramming protocols?
That's a very interesting question, thank you. How long do you expect transcription factors to be around? And particularly in relation to cell cycle. If they have such a short half-life that they're only going to be there in significant quantities if you put them in at a particular cell cycle stage, I would think it would be important. But if they survive more than one cell cycle, I think it would be hard to find an effect. So that's the way I guess. Would that fit with your understanding?
I mean, there are certain signals we can put in the transcription factors to be degraded at a specific stage of the cell cycle if you want, control their half-life. So that is possible.
Yeah. No, very interesting thought. Are you aware of any experiments where anybody has tried synchronizing cells? No. It's relatively easy to do. Synchronize cells and then use them at set times.